And our next speaker is Roberto Ripa from the Max Planck Institute for Biology of Aging.
Okay. First of all, thank you very much to the organizer for giving me the opportunity to share the results of our project today. And so we all know that dietary intervention like calorie restriction or intermittent fasting or fasting mimicking diet promote metabolic health and longevity in a variety of model organisms. However, we usually those studies start those interventions in relatively young animals during the young adulthood or middle age. So the question is, what happens if we start those interventions at this later stage when the animals are already old, are still effective? There are very few studies, and the take-home message is that all the animals seems to become refractory to such intervention, and in some cases, this intervention leads to frailty. And this is quite interesting, given that nowadays we know that there are other interventions, like rapamycin, for instance, that are effective even at the very late stage of life. So, now, if we consider those interventions, like, as I said, intermittent fasting or to some extent calorie restriction, they entails a cycle of fasting and refeedings. This is basically the core of those interventions. And then we ask a very simple question: does aging influence the fasting or the refeeding response? So in other words, is it possible that all the animals respond differently either to fasting or refeeding, and therefore change also the outcomes of those dietary intervention? So to answer this question, we use the short-lived turquoise killifish, and we characterize the transcriptional response either to fasting or to refeeding in young and old individuals in multiple tissues. And today I'm going to show you what we found out in the visceral adipose of this species. So this is just the experimental design is extremely simple. In this case, we fasted young and old animals just for 5 days. It sounds a lot of time, especially for the people that works with rodents. However, fish like humans, if you will, or other mammalia species can endure relatively long fasting period. Then we harvest the tissues, and then we perform bulk RNA-seq. Before to show the other data, I want to show you here the blood glucose levels. I just want to highlight that we really spent a lot of time to check that actually fed animals were indeed under feeding condition and fasted animals were under fasted condition. It sounds trivial, but you will realize soon that it is very important. So these are some of the results from the RNA-seq analysis. This is the PCA analysis, and suggested a reduced effect of fasting in the old groups. In line with that, we saw also a dramatic reduction of fasting responsive genes during the aging in these tissues. However, now things start getting weird. What we saw is a signs of anabolic resistance. So see, this is the gene expression profile of all the fasting responsive genes. And as you can see, many of them are not correctly reprogrammed during the feeding phase, suggesting indeed that there is a problem with the feeding response and not with the fasting response. So in other words, if you see this as a cycle, again, the fasting programs has to be terminated the moment in which the food is reintroduced in the body. But in the old, this reprogramming appear defective, and then some of these parts of this program runs over the feeding phase. And this actually associates with the suppression of energy metabolism and problems with lipid storage. Unfortunately, I don't have the time to show also this part of the data. So, of course, given this problem with the feeding phase, we checked the refeeding. And in this case, we did a temporal analysis of the refeeding response in multiple tissues in young and old animals. And once again, I will focus on the adipose. And I'm going to show you just the one data. This is the Kang pathway enrichment analysis. And you can see that young animals strong upregulate upon refeeding genes associated with energy metabolism, DNA replication and repair, and protein synthesis and proteostasis. However, this effect is blunted, is very mild in old animals, and also there is just a very weak response at 6 hours of refeeding, and then you see that 24 hours, strangely, all the animals upregulate pathways associated with inflammation and cancer. This was very surprising for us. And this indicated that indeed the refeeding response, more than the fasting response, is dysregulated during aging in this species. So, of course, we had many questions at this point, and of course, the most important one was, can we restore, can we find a way to maintain a proper refeeding response at later stage of life and try to see if this is enough to promote longevity or metabolic health? So we tried to go back to all our data set and check potential gene associated with refeeding. And we came across with this interesting regulation of the AMPK complex. So AMPK complex, as many of you know, is a gene master regulator of energy homeostasis by sensing energy nucleotides like ATP, ADP, AMP. So AMPK, in other words, can regulate metabolism, especially the ratio between anabolism and anabolism, by sensing the energy disposal. So the way how AMPK sense energy is through gamma subunits. Gamma subunits physically interact with energy nucleotides. However, as you can see, there are up to 3 gamma subunits, gamma 1, gamma 2, and gamma 3. This is true in killifish, mice, humans. Those subunits compete each other to bind the other partners, alpha and beta. Those subunits that fails to bind with the alpha and beta are degraded. So this means also that the relative expression of those subunits largely influence the final stoichiometry of the AMPK complexes. Now, what we found out is that for some reasons, fasting and refeeding differentially regulates the expression of those subunits. And this is regardless the length of the fasting phase. So always fasting downregulates gamma 1 and upregulates gamma 2. However, refeeding reverses this phenotype. Now, these are young, healthy fish. When we look in the old, this regulation appears blunted once again. As you can see in the dashed line, we have a chronic upregulation of gamma 2 and a chronic downregulation of gamma 1. So this is basically the model. It seems like that AMPK switch gamma subunits according to the refeeding phase. And this gamma 2 associates with the fasting and gamma 1 with the refeeding. And aging blunts this effect. So what's the relevance of these two complexes? And especially, what is the relevance in terms of aging and longevity in this species? So we decided to hyperstimulates both the AMPK gamma 1 complex and AMPK gamma 2 complex and study the long-term effect on the aging and longevity on these species. So when we did this in gamma 2, with gamma 2, we end up with old obese fish, despite this was associated with fasting. So we already know that AMPK is considered a pro-longevity gene, and in particular, I just show you that this is associated with fasting. But the chronic stimulation of this complex lead to obesity and insulin resistance. And surprisingly, this was very much in line with previous observation in mice and humans. Chronic activation of gamma 2 leads to obesity, hypertrophic cardiomyopathies, and even association with Alzheimer's disease. This was quite striking. And so we decided to check the effect of the activation of gamma 1. So once again, we generated a gain-of-function mutant, and then we studied the long-term effect on aging and longevity of this species. So what we found out is that those mutants retain a more youthful feeding and fasting response at the later stage of life. And this is the transcriptome of old mutants compared to old wild type. And we could see that basically those mutants have more upregulation of proteasomes, DNA replication, and higher energy metabolism. So these are the signature of the refeeding that I just showed a few minutes ago. So in other words, it looks like that the stimulation of this specific complex seems to promote the refeeding effect at the later stage of life. And with this, we also saw that the activation of gamma-1 indeed promotes more tissue regeneration. We quantify, for instance, the amount of FDU positive cells in different tissues, and this is the adipose, and you see that the mutants line actually tends to have higher number of proliferating cells. We also check many different parameters. I don't have the time to go through all of them. Overall, we saw a general improvement of lipid metabolism and glucose metabolism, and also we test the effect on longevity on 2 different transgenic lines, and in both cases, we saw lifespan extension. So to conclude this part, actually, we saw that the selective stimulation of the AMPK gamma-1 associated with the refeeding promote metabolic health and longevity in this species. To conclude, we actually saw also, what happens in higher species like mice and humans, and we found out that, in 2020, has been published this paper where has been characterized the fasting response in human subcutaneous tissues. And what they did, they took biopsies from before and after 24 hours of fasting. And what you can see is that the expression of AMPK gamma-1 and gamma-2 is indeed modulated during the fasting in humans. We also checked in the GTEx dataset, and we actually saw that in many organs, gamma-1 is downregulated during aging. And we saw this prominent downregulation, especially in the blood, which was quite interesting. And so we decided, in collaboration with the University Clinical Hospital of Cologne, to collect PBNC from all donors. So we had 100 older donors from 65 to 90 years old. We collected PBNC, and then we quantified the expression of the AMPK subunits. And then we correlate this expression with the multidimensional prognostic index, MPI, which is a sort of a frailty score that takes into account also other categories, like, for instance, the diet of these people, or how many drugs they take, or the social interaction. So, and overall, higher values of the MPI reflects poor health outcomes and higher mortality risk. What we found out is that among all these subunits, only the expression of gamma-1 seems to significantly anti-correlates with the MPI scores, indicating that healthier people tend to have also higher expression of gamma-1. So in conclusion, we believe that the selective expression of gamma-1 might induce beneficial effect also in humans and promote metabolic health and maybe also longevity. So with this, I conclude, and I like to thank Hans for giving me the opportunity to join his lab and work on this beautiful project, and then on Roma Müller and Maria Cristina Poriloris for the human samples and the analysis. And thank you for the attention.
Thank you very much for a great talk. We have time for a question. There in the back.
Yeah, thank you. Quite interesting. I have 2 questions, actually, if that's allowed. So first, can you actually feed those fish with something like high-fat diet, high-fructose diets similar to mice or humans? And does it matter what kind of diet you refeed them in order to change their AMPK response?
Okay, so in principle it's possible. We never tried, and we are using just a normal standard diet. So we never tried to see whether changing the diet during the refeeding it can indeed also modulate the refeeding response. This is definitely interesting, and it's something that we thought about, but we never did, yeah. But possibly, yes. Maybe if you reach of lipids or deplete, yeah.
Okay, thank you again for joining. And